My guest today is Professor Gina Poe, who is Professor of Integrative Biology and Physiology at UCLA. Her lab investigates the mechanisms by which sleep traits serve learning and memory consolidation. Welcome, Gina.
Hi, wonderful to be here. Thanks for inviting me.
Yeah, thanks for doing this. So you sent me a few papers. I found all of them extremely interesting. I didn't understand a lot of them, but I found them interesting. So I want to start with one of your older papers from 2006. Input source and strength influences overall firing phase of model hippocampal pyramidal cells during theta. Relevance to REM sleep, reactivation, and memory consolidation. Using simulation studies using a realistic model CA1 pyramidal cell, we accounted for the shift in mean firing phase from theta cycle peaks to theta cycle troughs during rapid eye movement, REM sleep, reactivation of hippocampal CA1 place cells over several days of growing familiarization with the environment. I've been talking to a lot of neuroscientists, Gina, and my ignorance is slowly moving up, I think. And so place cells, I remember, is sort of a marker for location, environment, and so on. And so, is it that your models here get familiar with the environment, and over time the firing patterns change? It's really fascinating.
Yeah, that's exactly right. Wow, you hit the nail on the head. This is a paper exploring how it's possible that we consolidate memories from the temporary storehouse in the hippocampus to the rest of the cortex, and then how that consolidation signal gets fed back into the hippocampus to tell the hippocampus that it no longer needs to store those memories in the novelty encoding pathways. So it can erase the novelty encoding pathways because it's already been consolidated. And so it's kind of it's a circuit-based signal that tells our brains that we no longer need to save things in our RAM because it's already been written to the hard disk. And so we can refresh the RAM of a hippocampus and allow it to be able to learn new things throughout the rest of our lives every day. Otherwise, our hippocampus would get saturated with these old memories. And so when we learn to become familiar with a place, for example, that map of the place, which is mapped in the hippocampus and continues to be mapped in the hippocampus throughout our lives, that map, however, is stored in a parallel and distributed fashion throughout the neocortex. And then when we return to that place, the cortex says, this book is over here, this door and window are over here. We're in this place, and the hippocampus says, oh yes, I recognize that. That's familiar. I don't need to remap it again. But if there's anything novel in that environment, the hippocampus can recognize through its novelty encoding circuitry what is novel, and then activate that novel circuit in with the familiar, and then during sleep sew that new piece of information into our old schemas. So this paper explores how that's possible on a circuit level, on an electrophysiological level, with that theta activity that happens in rapid eye movement sleep and also when we're learning, and then also with the neurochemical environment.
Yeah, this is so interesting for me, Gina. So, I do some work in AI, and the biggest problem in AI is discarding information. And so as new information comes in, we have to sort of value the new information and see if it is useful, right?
Yeah.
It appears that the brain has some sort of an optimization function that says, hey, I already know this, so don't worry about it, right?
Yes, that's exactly what this is getting to. And it's so exciting to me because so it had been known for decades that rapid eye movement sleep was important somehow for consolidating the things that we learned. And consolidation means writing from the RAM to the hard disk and, for a permanent memory structure, storage structure. But there had been a hypothesis by Francis Crick back in 1989 that said, well, maybe REM sleep is really for clearing the brain of all the things that we no longer need to remember, all of the extraneous information that we learned during the day that would just fill up our brain with crap. And so maybe we just need to erase. And that's why REM sleep is important for learning and memories, for erasing it. Well, sleep researchers who had done these decades of research showing that REM sleep is important for consolidating memories said, oh, Francis Crick, his area is different. What does he know about sleep? And it was a hypothesis, but it didn't propose an experiment or a test. And so in the late 90s, when I got to the University of Arizona, I knew they had a way that I could test that hypothesis. Is REM sleep for learning or is it for forgetting? And so I was really excited to do these experiments where I could see whether the hippocampus fired during REM sleep in a manner that's consistent with strengthening memories or for erasing memories. And what I found was something that I hadn't even guessed, which was that the hippocampus was active in a manner that's consistent with strengthening new memories that had not yet been consolidated and weakening old memories that had already been consolidated. So in the same REM sleep period, the hippocampus was activating these different memories in opposite phases of the theta cycle to strengthen and erase, dependent on the consolidation state of that memory. And so I spent the next 6 years trying to figure out how that is possible, because there is a set neurochemical environment during REM sleep. There's the set pattern of theta activity. And how is it possible that the hippocampus can distinguish between memories that have been consolidated and memories that hadn't? So I explored it with a mathematical modeler, Victoria Booth, who had worked with theta activity in the hippocampus slice in a laboratory in New Jersey. And she had just come to the University of Michigan, where I was at the time. And we started a conversation and we started exploring papers on it. And she was the one to tell me that theta phase was opposite in 2 different layers of the hippocampus. And then we looked at norepinephrine and serotonin levels and how that changes with REM sleep, and then receptors in the hippocampus. It's just so elegant, complex at the same time as just beautifully elegant. So maybe AI could learn something about, having the system automatically report whether or not something is familiar and then automatically erase if it is.
Yeah, it's a true optimization problem, right? Given a limited capacity, the objective function is how do I best utilize that capacity?
Yeah.
And there's a lot of extraneous information that we pick up all through the day.
Yes.
And so if I understand this correctly, Gina, what you're saying is that during the REM sleep, there's some sort of an optimization program going on that says, yeah, this information is important because I don't have it stored, so I'm going to store it. This information is not useful because I already have it, let's say. That's sort of item B. And item C might be that, yeah, this information is interesting, but I don't necessarily have to store it. I saw a cat at the 1 p.m. Outside the door. Yeah, it's interesting, but do I really have to store that information?
Exactly. Right. And so what we don't know yet, what's still a complete mystery, is how we tag the things that we want to store and don't tag the things that we don't. Or maybe the opposite. Maybe we tag the things we don't want to store and tag it for erasure, mark it for erasure. We don't know the answer to that yet, but I do think it has something to do with the attention systems of acetylcholine and norepinephrine, maybe even serotonin, and the receptors and what happens inside the cell when these things are active, because the noradrenergic system, the noradrenaline system, which is like adrenaline, but it's noradrenaline for the central nervous system. And that gets activated whenever something is novel, and we switch our attention. It helps us switch our attention to pay attention to it, adds acetylcholine to that whole mix, which helps us to learn quickly. So it might be that when something is relevant and we can tell it's going to be important, we don't know exactly why necessarily it is important, but it seems like it's going to be important that we add these tags, which are added through norepinephrine. And there have been some great research by other people showing that these tags might be related to the things that happen inside the cell when something new comes along.
Yeah. So if I think about this sort of a decision tree perspective, so I could first recognize whether it's novel or commonplace, let's say. That's
Right.
the first node in the decision tree.
Yeah.
If it's commonplace, maybe I don't want to spend too much time on it. So I go on the novel track. Then I say, do I already have it stored? Then I don't have to worry about it. So it's novel, I don't have it stored, then I have to worry about it.
Yeah.
So is it as simple as that? I'm making it sort of mechanistic in my mind.
No, I think it is as simple as that. I think it involves at least 3 different neurotransmitter systems. It involves at least 2 different circuits that come into the hippocampus. But I think it can be as simple as state and the anatomy, the way the circuit is set up. Yeah.
So we're going to get into this in the later papers, but so if you don't have REM sleep, then the brain is so there's garbage. There has to be garbage collection, so to speak. Right? There's garbage sitting there, and the brain hasn't sort of sorted through it. Is that the way to think about it?
That is the way to think about it. Yeah, I think of wakefulness as kind of a giant party that the brain is having, and your guests in your party are bringing gifts to you. It's like a housewarming party, maybe, and that's an analogy. If something traumatic happens, it might be like a guest has brought a bull into your party, and it's doing all kinds of damage, and then what you have to do is once the party is, all the guests are gone, is you need the cleaning crews to come in and sort out what are gifts, what is trash, and what do you need to put away, and how do you put it away within your home so that it makes sense and not just put it in random spots. And that all happens during sleep when the party guests are gone. And if you've had a traumatic memory, a traumatic event, you need to lead that bull out into the backyard rather than wreaking havoc in your house, and that requires special work and a way to sort things out. If you can't do that during sleep, then that bull stays in your living room. That is not a good thing.
Yeah, so I think we're going to talk about this later, Gina. So so the traumatic memories, it creates a lot of stress to the brain, and so there is an objective to sort of either forget it. I don't know what the right methodology might be, or utilize it less in some way. So that's also happening during sleep, right?
Yes. So that's the current hypothesis that I'm working out with researchers around the world, Australia, the Netherlands, and the United States. The idea is that stress increases the amount of this norepinephrine, this noradrenaline neurotransmitter that I talked about. And it's really important that that noradrenaline level be 0 during REM sleep, which it normally is. But if you get REM sleep that's too soon after the traumatic stress, when the producer of noradrenaline is still excreting tons of it, then that sleep becomes maladaptive. And instead of weakening those aspects of the memory that need to be weakened. I mean, you don't want to forget the trauma, but you want to weaken the emotional parts of it at least, so that when you remember the trauma later, you don't have to recall all the fight or flight, sensations, that your body doesn't have to go through all of that again. You can remember the semantic facts of it. You can incorporate that into the schema of your life and the episodes, but you don't have to tie into that memory all of the emotional aspects. And I think if noradrenaline is still present during REM sleep, you cannot weaken those synapses that need to be weakened. And instead, you continue to strengthen as though it's happened yesterday throughout the rest of your life until you can break that cycle. So one of the abilities to avoid post-traumatic stress disorder might be to avoid REM sleep altogether until your noradrenergic system has calmed down and you're able to find some peace, make some peace. Okay, and in the future, I'm going to avoid this way. Find a way to come to a place of peace so that that noradrenergic system is not any longer telling you to fight or flee. And instead you can breathe and relax. And then when you go into sleep, it becomes adaptive and does what it's supposed to do, weakening the emotionality from the facts.
I mean, this has a lot of implications for sort of therapeutic interventions too, right? So if I understand you correctly, Gina, what you're saying is that if you have a PTSD type event and then you have REM sleep soon after that, it actually it's sort of the negative way in some sense, right?
Yeah.
Yeah, that
It
is.
strengthens the trauma instead of weakening it. The
It
trauma.
reinforces the trauma instead of weakening it.
Yeah.
Yeah, my tendency, if I have a lot of stress, I typically say I'm going to sleep a little bit.
Yeah.
Yeah, but it has to be REM sleep, right? It has to be REM sleep for it to
It has
matter.
to be REM sleep, and so it might be that it would be good to have slow wave sleep to release growth hormone. Gets released really strongly during slow wave sleep. It clears out debris. It does a lot of good things. But if you go into REM sleep while your locus coeruleus, which is a source of noradrenaline in your forebrain, is still active, it would be bad. So it would be a great therapeutic intervention. A biomarker, for example, would be great to develop to tell whether or not your sympathetic nervous system is still too active while you're sleeping. So it lets you go into slow wave sleep, but if it sees you slipping into REM, it says, no, your sympathetic system is too high, you need to wake up. Or it gives you a drug or something that helps your sympathetic system be not listened to while you're in REM sleep, so your REM sleep can be adaptive. I think this is a really good target for therapeutic interventions, and I think it would be good even if people have had PTSD for years. I think you could still do this adaptive work in sleep if you're able to modify your sleep after you've recalled the trauma.
Yeah. I mean, the beauty of this observation is that um, it has a negative effect, meaning, so if you have a bad event and you go to REM, it reinforces the bad event, right? That's what you're saying.
Yeah.
And so in some sense, you have to keep people away from REM post the PTSD type event. And I mean, you have a very small window there.
A very small window.
So I'm thinking, somebody dying or something like that, and but that is you can intervene within that window.
Yeah.
And so it is actually good for people to talk to somebody really, sort of suffering. Don't let that person go to sleep, so to say.
Let them work it out first. So
Yeah.
the problem with talk therapy right after a trauma is that sometimes it just continues to keep it fresh in the person's mind, and the stress remains for longer than it would otherwise. So that's actually counterintuitive, or counter what you want to do. So if you talk with someone, which I think is a really good idea, talk with them to help you find that place of peace, to help you resolve, what did I learn from this? If there is something to learn, how can I avoid it in the future? How does this fit in with my life? And how does my worldview, if you have a worldview that would help you, analyze this trauma and come to peace with it, then how does this thing fit in with my life and my worldview? That's all a good thing to do, whatever helps calm down your sympathetic nervous system. And for some people in some traumas, it might be best to deny instead, right? Maybe one way to calm down your nervous system is to just kind of put it out of your mind and pretend it never happened, and that can be a very powerful tool as well. You probably have to face it someday, but hopefully at a time when you're more ready to face it. But if you can face it now and you can come to a place of peace within the first few hours of the trauma, then wonderful. Then that would be a great thing to do.
Is there sort of a chemical intervention that prevents REM?
Oh yeah, lots.
Okay.
Lots of things prevent REM, including alcohol. So
Okay, yeah.
one of the things that I heard that British soldiers do, and after a buddy has, experienced a trauma in the theater of war, is they get them drunk afterward, and that might not be such a bad idea if it prevents them from going to REM sleep, even if they do go to sleep for a certain number of hours. I would not recommend it forever, but maybe that first night, being drunk would be not a bad thing. But there are a lot of other things that prevent you from going into REM as well, including just staying awake, right? And one of the things that neurologists often, or physicians often prescribe if someone's been through a physical trauma, especially if the physical trauma could affect the brain, like be a concussion or something like that, is they recommend that you stay awake for a certain number of hours. That might also help prevent that trauma, from the psychological trauma, from being overwritten into your memory structures. So
yeah, we could talk about this forever, Gina. I want to go to some of your other papers. So 2016, we have another paper, so different simultaneous sleep states in the hippocampus and neocortex.
Yeah.
You said we found that hippocampus and neocortex independently progress through sleep-waking states.
Yeah.
Challenge the utility of single EEG, electrode, and purely behavioral characterizations of states, as well as canonical views of sleep as a global phenomenon.
Yeah.
We suggest that electrode location affects the scoring of states, with implications of prior sleep and sleep deprivation studies.
Yeah.
So it sounds a bit like the brain is sleeping in different parts at different times,
Yeah.
and you cannot really say the brain is sleeping, right?
Right. Yeah. So it used to be thought that the brain slept homogeneously. Then we learned that in certain mammals, like who are at sea, like whales and dolphins, who have to breathe, they have to come to the surface and breathe, that they can sleep unihemispherically. So one hemisphere can be asleep while the other hemisphere is awake. It was never known, and still not known, whether or not that one hemisphere that's asleep can go into REM sleep. But we do know that every animal that we've been able to do the measures of probably have two states of sleep, including REM and non-REM sleep. And so we still don't know the answer to that. But that was the first indication that it's possible that part of the brain can be asleep while another part is awake, at least. Then we learned through some research done in Switzerland that particular areas of the cortex can be in deep slow wave sleep, while other areas of the neocortex kind of don't look like they're in deep slow wave sleep. So the amplitude of slow waves and the presence of slow waves can appear in some areas and not others. And that can happen more when you're sleep deprived. Even when you're awake and doing a task, there can be areas of your brain that look like they're fully asleep. Have you ever seen someone that's kind of zoned out? That's probably what's happening, is parts of their brain are asleep while other parts are awake. And but still, those are all non-REM versus waking kinds of distinctions. What we've never seen before until this paper in 2016 is that portions of your brain can be in the dream state of sleep while other portions are not. And this happens normally. And this is in rats. We did a study in rats, and another Ernesto Duran did the study the next year and found the same thing. He was doing a study in Germany. But also in rats. So we still don't know if this is also true in humans. However, rats and humans share so much commonality in so many other realms, it's quite possible. So actually right now, one of my graduate students is looking at some human sleep data with electrodes implanted deep in their brain versus in the cortex. And yeah, I'm excited to not report yet, but I think it's going to be really, really neat to see.
Yeah, I mean, it makes sort of an intuitive sense from an evolutionary perspective, right? So predation would have been a major threat for animals and humans. And so it makes sort of sense if you can, let some parts of the brain, sort of attentive when the other part sleeps, I would think.
Yeah. And, and there are dissociated states that are kind of not fun, like when you sometimes wake up out of REM and your whole brainstem hasn't released your muscles from moving yet, so you're paralyzed. That's no fun, and it's kind of scary. That's a dissociated state. There are dissociated states where sleep intrudes into wakefulness. It's called confusional arousals, and that's no good either. So these dissociated states can be dangerous and bad. You can have REM sleep without atonia, where your brainstem isn't in REM sleep, where your forebrain is dreaming vividly, and so you can act out your dreams. So it's better to have everything proceed in a very lockstep manner. However, it might be that some of these states, when your forebrain is in slow wave sleep and your hippocampus, say, is in REM sleep, it happens quite normally. It happens even more often in animals that have learned something new. So it might be that it's actually doing something unique, special, to have this dissociated state. And it might be very necessary for consolidating memories or integrating new things into old information. We don't know the answer to that yet. That's an area.
Yeah, so it has nothing to do with energy, right? So, so in computers, for instance, we have a way to get to a computer. So it has a CPU, then gets stuck sometimes.
Yeah.
So we have to sort of go a different way to restart the computer. Neuroscience hate me because I sort of make some connections to computers. It's nothing like the other. So it seems to me that, so there's some objective in terms of REM, all the stuff that we talked about before.
Yeah.
But either there is some danger, some risk that you need to worry about, or you need some sort of a reset mechanism if things are not quite what you thought is going to happen.
Yeah.
Is this sort of a reset possibility?
Yeah. So one of the cool things is that throughout sleep, our forebrain, orbital frontal cortex, prefrontal cortex, all of that is really off. I mean, metabolically, it's really low in activity.
It's done its work already. It has done the work. The old day it was working, and now I say I'm gone. I'm done.
Yes, and I'm done. And if you're sleep deprived, your prefrontal cortex has to work twice as hard, involve many more areas in order to accomplish the same tasks. So that off state seems to be really important. The one time during sleep when that prefrontal cortex is on is during a transitional state when the hippocampus is firing bursts of activity coincident with reactivating memories, and the prefrontal cortex responds and reverberates in a sleep spindle mode. Sleep spindles are about 1.5 second long waves that are coordinated between the thalamus and the neocortex. The hippocampus, the thalamus projects to the hippocampus back to the thalamus to the prefrontal cortex, and somehow during these brief periods of time, the prefrontal cortex is really actively responding to the hippocampus, and that might be when we're sewing new information into old schema, but it's brief, and so it might be these dissociated states are when this happens, that it's brief, but it's super powerful. It can rewrite our memories, it can change our minds, it can change the way we see the world, and so it might be a really important time of maybe it's the reboot. I don't know exactly how you would have the analogy in the computers world, but it's a time when you're rewriting your what is it called, kernel or something.
Right. Yeah. So would you say, Gina? So the hippocampus is sort of working through all the data. And trying to determine which one is more important to store and which ones are good to discard. But it may not have the sort of the algorithms So it has to go to the, sort of prefrontal cortex to say I got bucket A, bucket B. Which, which one do you think I should store?
Yeah.
Do you think that's happening?
Yeah, we don't know how, but one of the things that we are starting to discover is that the brain is a lot more modular than we imagined, especially the acetylcholine, which is the attention sort of neurotransmitter. That system in the basal forebrain, the bottom part of the forebrain, was thought is a pretty small cluster of neurons, and it projects all over the brain. And it was thought that when it was doing one thing, the whole brain was in that state. When it was doing another thing, the whole brain was in that state. We now know that there are modules of attention, so that it's not like we just overall are more attentive or less attentive. That we put acetylcholine in those areas of the brain that need to be doing that task. And other areas of the brain don't have as much acetylcholine, that there's a structure to that. And it's the same with this noradrenergic system I talked about with stress and attention that's also modular. It's a cluster of, in the rat, it's like 3,000 neurons on each side. And that's very, very small. It's a very small area. So how could it be modular? But it is. Somehow it's still modular. And Dan Chandler and others in Pennsylvania have been showing that.
Yeah, I can see the prefrontal cortex, saying to the hypothalamus that, I thought I was done. I thought you got this.
Yeah.
And then it comes back and say, I got bucket A, bucket B. Which one do you think is more important? They say, I did my work all day.
Yeah.
Now you can figure this out yourself.
Right. No, they work together. Thankfully, they work together. The hippocampus can tell the cortex, these are the new things. I know most of this is familiar, but look at these new pieces of information. Could you fit that into your schema somehow? And I think during sleep, and only during sleep, when noradrenaline is not present, is when the prefrontal cortex can loosen the schema to fit in the new piece of information and discard other pieces of information that no longer should fit into that schema.
So do we see activity in prefrontal cortex, fairly high levels of activity during REM?
So again, prefrontal cortex is off during REM and it's off during slow wave sleep. And the only time it seems to really be active in coordination with other areas like the hippocampus is during these sleep spindles. And so, yeah, that's at the transition from non-REM to REM sleep. It's a very transient state, a unique state in terms of neurotransmitter levels, totally unique in terms of the presence of these sleep spindles. Sleep spindles are really cool because your density of sleep spindles correlates well with IQ, your intelligence quotient. So, and your ability to learn across sleep. If you're able to increase whatever baseline level of sleep spindles you have, if you've learned something new and need to incorporate it into your forebrain, your density of sleep spindles goes up. And if it doesn't, you don't incorporate it. So
it's not a transition between REM and non-REM, you said, right? Yeah. So that's where sleep spindles are active.
Yes.
So in some sense, so if you have a lot of density, a lot of activity there, it's basically, I don't know what the right term would be, but the hippocampus has sort of a way to get to the prefrontal cortex and get answers very quickly.
Yes.
So that it can make whatever decisions it has to make.
Yeah, they're reverberating together during those sleep spindles. It's really, really interesting. Because we used to ignore sleep spindles and that transition state because it was so short, it was so brief. People didn't think it must have been very important. It was just a transition from one major thing to another. But in fact, it might be that it's so brief because it's so powerful. You really don't want to be rewriting your entire personality and all your schema every night. You want to only do it for those things that are necessary and only as much as is necessary.
Right. So in this paper, you have a rat study where we're looking at measured electrical activity and score sleep-waking states directly from the brain structures of interest rather than interfering states of neocortical electrical activity. So, I mean, rats are quite similar to us.
Yeah, they are.
And so what's sort of the major conclusion from this paper?
So the major conclusion is, first of all, we shouldn't ignore anything. And in fact, there is no, useless transitional state, that everything is probably important. And that we really need to be measuring sleep from the structure that we think we want to test. For example, if sleep is for learning and memory, you can't just expect to slap a couple of surface electrodes on the visual cortex and assume what the rest of the brain is doing from that. You really need to be recording from those areas of the brain that are involved in learning and memory, and where it is that you want to be writing those memories. So you need to be recording from those structures rather than, and that's problematic because in humans, electrophysiologically, we can't see very deep with these surface electrodes. We actually have to implant them deep, which is dangerous. It involves surgery and, the risk of infection and all of that. So we still need to use animals, I think, to get at some of these questions. And then those precious few people who do have depth electrodes implanted for whatever reason it was needed to be implanted, we need to, really mine the data from that to see how much can we translate what we're finding in rats and cats to humans.
So you said the very highest level, you say consequently electrode location affects estimates of sleep architecture, state transition timing, and perhaps even percentage of time in sleep state. So what you're saying is that we can't really just get data from certain locations of the brain and make conclusions. Because it seems like some parts are sleeping, some parts are doing work, and then the ones that are doing work goes to sleep, and other parts wake up. So it's sort of a very complex modular architectural system.
I think that's what you want to call it, modular. Yes, and it's problematic because with sleep deprivation studies, for example, if you want to see, is REM sleep important for memory? You can wait until someone goes into REM sleep, wake them up, and assume you cannot assume from electrodes in one area that you're waking up the whole brain and disturbing REM sleep in the whole brain. It could be that the hippocampus can go into and out of REM sleep without the cortex ever registering REM sleep at all. And so it might be the hippocampus is getting 3 times as much REM, and under certain circumstances, like an intensive learning period, than the neocortex is getting. And so, if you don't see an increase in REM after a certain learning task, you can't necessarily say, based on neocortical studies, that REM sleep is not important for consolidating that task.
Yeah, I often think about, the Pink Floyd song, "There's somebody in my brain, it's not me." It seems like there are a lot of people in the brain.
Well, you know what? It's interesting you should say that, because the first time I saw a video of neurons migrating to where they're going to go in the end during development, it really looks like you're looking at slugs making decisions about where the food is or something like that. It extends a filopodia this way and it retracts and extends it this way and then this way. And then it says, okay, really, this one's the best. And the whole neuron follows. So it's making decisions. So it's almost as though your brain is filled with billions of individuals making decisions, talking to one another. It's almost like our brain is a city, or maybe a universe full of communicating entities that do different tasks. Different parts of it do different. There's those that take out the trash. There are those that are lawyers, or whatever. There's a whole city that needs to work together. So I don't think there's one other person in your brain. I think there are billions of
one could argue that every neuron is sort of like a person. It might have a personality. The hysteresis it has gone through over time sort of defines its personality. So how it will fire given information also changes over time. So it is mind-boggling.
It is mind-boggling. There are those who believe that the seat of consciousness is in the microtubules, which are in every neuron, in every cell, actually. It determines its shape and where it will go and what it connects to. I don't know. I don't know what the seat of consciousness is. It shifts every time I do a new study.
Yeah, it's something that humans attach a lot of importance to. I'm not sure. I mean, we say only, a lot of people who've done a lot of work in consciousness say that only humans have consciousness. And they kept asking, how do we know this? I mean,
We don't.
I have,
And I don't believe it. I don't believe it for a second.
yeah. I mean, one
Not
could go
even
all the way to the other
self-conscious.
extreme. Sorry.
Yeah, I mean, even self-consciousness, which is thought to be the highest form of consciousness, conscious that you're conscious and conscious that you're separate from another individual. You look at dogs, they can be very self-conscious, they can be ashamed, they can be embarrassed. Cats can definitely be embarrassed. And, I definitely don't believe that that's what we should base we shouldn't hang our hat on that, because we don't even know what consciousness is. And even when no matter which way we define it, we can find other animals that have some form of it.
Yeah, I mean, there's a huge definitional issue. There's a huge sort of theory of mind issue. We don't really have a theory of mind yet. So we can make up anything, and it wouldn't let us go anywhere. I want to go into another paper from 2018, Unraveling Why We Sleep. A quantitative analysis reveals abrupt transition from neural reorganization to repair in early growth. So you said sleep is known to serve disparate functions, most notably neural repair, clearance, and reorganization. Yet the relative importance of these functions remains hotly debated. Here we create a novel mechanistic framework for understanding and predicting how sleep changes as individuals grow. This was really interesting for me. So I don't know if it's this paper or the next paper, sort of the two different things. One is sort of the temporal relationship on humans, how it changes. Another is sort of the size of the animal and sleep. Is that in the
Yeah.
same paper? I can't quite.
Yeah. So the size of your brain, and we studied sleep records from several different species, and as well as humans across the lifespan and other species, that other people came up with the data. We just reanalyzed it, and we reanalyzed it in relation to brain metabolism and the events that we knew were happening during development. Synaptogenesis, the creation of synapses between neurons, and synaptic pruning, and just basically how much energy the brain is expending in order to do what it does. So the lessons that we learned, there have been other great studies showing that sleep was or was not related to metabolism across species, and sleep changed across development. So ontogeny and phylogeny, sleep changes. But this paper sort of unites all of those studies under a common set of calculations, which really show that the function of sleep probably changes from the time when we're early in developing in our brain. The overall main function of sleep probably changes from the time before we're about 2.5 or 3 years old to afterward. And in those first 3 years, any developmental psychologist or neuroscientist will tell you that our brain is really forming those schema and figuring out, how to learn, and, who our parents are, and, how to walk and how to talk, and what our language is versus all the other languages, and, what parts of our nervous system that we're born with. We're born with a well-connected brain that can really encode almost anything. We need to prune away a lot of that as we develop, because, for example, if you're born without the ability to see, your other areas of your senses will need to be able to take over all that brain space that would otherwise be devoted to vision, right? We don't want to just waste all that brain space. We want to use it for the other senses. And so our brain rewires based on the experiences into which we were born, and those first 3 years are critical for that. They're also critical for language learning, It sets us up for ability to do well in school for the rest of our lives. Emotionally, how we relate to others, what happens to us in those first 3 years, how our parents relate to us, whether or not our needs are met, how loved we are, all of that sets us up for the rest of our lives. So sleep in those first 3 years is really critical because sleep is the time when we are pruning away things that are no longer necessary to be encoded. So we're born with kind of a omnipotent brain that gets specialized and adapted to the environment in which we learn, in which we're born. And so sleep is the time when that happens. And then after we're about 2.5 or 3 years old, we switch into a mode where we are fitting the things that we learn into those schemas. So it's not like, so REM sleep and that transition to REM sleep spindle state are the important times for pruning away things that are not important. You'll need that for the rest of your life because things will change, but you might need it less than you needed it in those first 3 years. And what takes over to be more important is maintaining those synapses that you've now decided you need to keep. And so cleaning away things that build up as we are interacting with our world. And that's slow wave sleep. That's the slow wave sleep state.
Yeah, this is not the paper, Gina. I want to get your perspective on this. So it has a lot of policy implications, right? So we know that the first 3 years are significantly important for a baby.
Mm-hmm.
And to return to society, to really investing in those 3 years
Mm-hmm.
are close to infinite. So I mean, you can think of all sorts of things like crime, IQ,
Mm-hmm.
taxes society will get, let's just call it taxes, for the rest of that baby's life.
Yeah.
There's so many things that society could influence in the
Yes.
first 3 years by
Yes.
investing very little in
Yes.
those first 3 years, right?
Yes.
But it doesn't look like we have figured it out.
No, I don't think we have. And one of the things that we have not done is trained parents how to parent, because it is parents who are the most time with the babies the first 3 years. It doesn't matter if the parent is biological or not. It's just whoever's spending, the caretakers, those people need to be trained on what to do and what not to do, how to set your baby up for a lifetime of emotional health versus disability. And so we need to spend a lot more time training parents how to parent. We'd spend a lot more time teaching people how to drive a car than we do and how to make a person, you know? So I totally agree with you there.
Yeah, making a person is probably much more complex than driving the car, but we spend nothing, almost nothing there.
Mm-hmm.
So I want to finish up with the latest paper, shining a light on the mechanisms of sleep for memory consolidation. So it's a paper reviews all optogenetic studies that directly test various sleep states, trait, and circuit level activity profiles for the consolidation of different learning tasks. And recent findings inhibiting and exciting neurons involved either in the production of sleep states or in the encoding and consolidation of memories reveal sleep states and traits that are essential for memory. We talked a bit about this. So um, just to put this in a more detailed context, you talk about REM sleep, non-REM sleep, NREM,
Mm-hmm.
and the N2 transition to REM.
Mm-hmm.
And so sleeping is not just sleeping, right? Sleeping is
Yeah.
very different types of things, isn't it?
Yeah, lots of different things. And your brain is busy doing what it needs to do the whole time. So this is one of the reasons why we shouldn't sleep deprive ourselves, is because really important work is going on. It's not like you're just leaning and loafing and not doing anything. You're really, you look at a teenager sleeping in on the weekends, and you don't want to say they're lazy, because in fact, their brain is doing really important things during that sleep state.
Not for the teenager, but most other people, yeah.
Yeah. Yeah, so teenagers, there's a lot going on in their brain. They're still changing quite a bit. Prefrontal cortex is forming, and they need just as much sleep as preteens. But there's a whole host of environmental and physiological factors that push their sleep a little later into the night and into the day. So it really is good for school districts to delay start times for teenagers because there's a whole host of factors that push their bedtime to later. And then if you keep the same school time, then they're getting less sleep. And sleep deprivation is really, I think, we know it's a bad thing for emotional control and for learning. So
where do you stand on this sort of, it has become quite fashionable to get power sleep, like 20 minutes sleep in the afternoon. I think the Spanish had figured this out a long time ago.
I actually think, yeah. Go ahead.
Where do you stand on that?
Yeah, I think naps are fine. And there are people who hate to nap because they always wake up really feeling bad and they don't get anything out of it. And I'd say don't nap then. If you don't like it and doesn't do anything for you, don't do it. There's a great series of studies by Sarah Mednick that shows a nap it can be as good as an entire night for consolidating your memories. But she also found later that those who do not nap ever because they hate it, nap is not as good as a whole night for them. It doesn't do anything for them, and in fact, makes them feel worse. So follow your body. I think if your body says take a nap, take a nap. The only thing I would caution against is taking a nap too late in the day because, for example, if you take a nap at 5 p.m. and you won't expect to go to bed at 10, well, that nap, if it's long enough especially, can really reduce your homeostatic pressure to sleep so that at 10 p.m. or 11 p.m. or 12 p.m., you don't have enough steam built up to force you to get into that sleep state. So it might disturb you for the rest of the night, and then you might get into a weird sleep waking thing that's outside of the normal circadian rhythm cycle. So you want things, timing is everything. It really is. So timing of a nap, I would say earlier is better in the day. So if right after lunch you feel like you need to consolidate all those things you learned in the morning, you're in college, you need to take a nap, go for it. I think it could be as good as an entire nap for consolidating things. And 20 minutes, the cool and magical thing about 20 minutes is that it's not long enough to get into that deep, slow wave sleep that cleans your brain, but it is long enough to what has been shown is the adenosine, which builds up as ATP is broken down when our brains are active. Adenosine triphosphate is the power structure, the fuel for our brains, for all of our cells. And it breaks down as we continually use our brains to adenosine diphosphate, then monophosphate, and then adenosine. And so that levels of adenosine build and build and build in our brain the longer we're awake. So what happens in the first 20 minutes of sleep is adenosine somehow gets beautifully transitioned back to ATP. So in terms of a power nap, it literally is a time to rebuild the power in our brains and all over our bodies. So it's really a neat thing. The other thing about adenosine is that is the neurotransmitter. That's the substance. It's not really a neurotransmitter, but it's the substance that is blocked when you drink caffeine. So coffee, tea, whatever has caffeine in it, the receptors for adenosine are blocked. So you can fool your brain into thinking that you've had just woken up from a power nap because the receptors are blocked and can't see all the adenosine still swimming around in your brain. But coffee can't do what sleep can do, and that is convert adenosine into ATP. So it can't restore the power needed. It just fools your brain into thinking you're refreshed.
Oh, that's beautiful. So if I understand this correctly, Gina, so if I think about sort of a 2x2 matrix, some people benefit from naps, and apparently some people don't, whatever the reasons are. But on the y-axis, is it a function of how much information is coming in? So you talked about students, for example. Do students benefit from a nap more than, let's say, 60-year-old who not a lot of information is coming in?
Well, so that's the thing. Adenosine builds up during wakefulness no matter what you're doing. So it's just the fact that you're awake, it just expends a lot of energy and breaks down a lot of ATP. So even if you're just sitting in a rocking chair on your porch, you're thinking you're thinking there's things going on. So what intensive learning does is it increases the homeostatic drive for REM sleep and that transition to REM sleep state. So so we get great studies by Subha Maldada has shown that we need and we require and get about 300% as much of that transition to REM with all those sleep spindles. There's also something called P waves, these big glutamatergic surges during after an intensive learning period. And if you block that 300% increase, you get the inability to incorporate that new information into our old schema. We get probably up to 50% more REM sleep time, which is a lot, but it's less than 300% more of the transition to REM. But anyway, those things are homeostatically regulated by how much we are learning. So, but a 60-year-old can learn a lot too. So, a 60-year-old can learn a language, and they will need just as much of those other states of sleep as a 20-year-old. Yeah.
That is good to know. So, so in conclusion, Gina, this seems like an area that is still developing a lot. I mean, we learned a little bit about the brain, but we still have a long way to go.
Really?
So graduate students getting into this area, I think they benefit a lot, right? I mean, it's a hugely important area. We didn't talk about this in this one, but I suspect sleep also has some connections to a lot of the neurodegenerative diseases, perhaps Alzheimer's, and we are all, kind of living longer and expect to have some neurodegenerative disease in the future. And so there seems to be a lot here that students could really get into, right?
Oh, there's tons of mysteries left, and really exciting time to be getting into neuroscience and to sleep research, because the generation that is starting to retire now have laid a fantastic foundation, but they have not solved it and they haven't figured it all out yet. It's just a great foundation on which we can all, build to find the answers to these really important questions. Alzheimer's is one of them. Parkinson's.
Excellent. Yeah, this has been great, Gina. Thanks so much for spending time with me.
Thank you so much for inviting me. It's been a pleasure.
Thank you. Bye.
Bye.
Gina, if you're still on, you can